Hello everyone, welcome to our second lecture this week. Hopefully by now you watched that video that I had for you guys. It's a really interesting one with Bonnie Bassler. I was lucky enough to actually get to see her give a presentation here, and the woman's brilliant. And she's also an excellent speaker, which doesn't always happen with faculty. But what the purpose of this lecture is to kind of give you an idea of how humans have also influenced bacteria. But before we talk about that, I want you to realize all the different amazing things bacteria do, okay? Now, I do expect you to know information from that lecture, all right? Mainly at the very end, she lists off 4 things that she wants you to know. You are responsible for those 4 things. So go back to the video, okay? It starts off with bacteria communicate through quorum sensing, and then 2, 3, and 4. I can't remember what order they're all in. But anyway, make sure you write those down because you're also going to see that on next week's quiz, okay? So relax, just go watch the video, list the 4 things, you'll be fine. So let's talk about how antibiotic resistance happens. And by the way, this is 100% totally and completely caused by humans. Yay! Once again, we've managed to mess things up a bit. Is anybody here surprised? I'm not. Just kidding, sort of. Anyway, let's say we have a bacteria. It's H. pylori is one particular species. And if this bacteria is a normal representative of its species, it produces an enzyme. And the antibiotic will target that enzyme production center and knock it out, and then the bacteria dies. Okay, this is when you first have a bacteria. Eventually what happens, though, and when enough bacteria grow, just by random chance alone, someone's going to be a mutant to where they don't produce that enzyme and they don't need it. Okay, so even after you add the antibiotic, that guy survives. In fact, he lives and then can reproduce and create more bacteria that are also mutants not producing the enzyme, and this is how we get antibiotic resistance. Yay! Okay, and honestly, this is something that we should be concerned about. And so a lot of bacteria that cause disease are starting to have antibiotic resistance, which means you can take as much antibiotics as you want, it's not going to kill them. And then the only thing left to save us is our immune systems, okay? And if you're immunocompromised, if you're older, if you're diabetic like me or my husband, those are definitely things you have to worry about, all right? So just keep that in the back of your mind. So whether we realize it or not, actually, humans have been using antibiotics for tens of thousands of years. We know this because certain antibiotics, like tetracycline, if you take too much of them, they will start to deposit in your bones. In fact, you will have stripes on your teeth if you have too much tetracycline. All right, so we know that back in the day, humans were munching on plants that had tetracycline, an antibiotic in them, and that kind of makes sense because people have been getting sick for as long as we've been around. All right, there's always this evolutionary arms race where we try to kill the bacteria that make us sick, and the bacteria try to bounce back, and that back and forth, and that's how that has happened. So for at least tens of thousands of years, and that's not a specific date, I know, but for a very long time, we've been taking antibiotics to kill off and ward off disease. Ancient Chinese cultures, which have been around much longer than North American culture, and many of the other cultures have also been using antibiotics that come in plants, okay? So plants and other bacteria and fungi all have, antibiotic properties, and so certain cultures have made use of that for a really long time. The good news, then, their people live. Whatever cultures use them, but bad news, then, this is how you get antibiotic resistance. So kind of a double-edged sword. So let's give some overall basics on bacteria. Okay, so the bacteria are really phenomenal little creatures. We don't give them enough credit. They have 2 different sources of DNA. They have their bacterial DNA, and this is where they get instructions to basically make proteins and do their bacterial thing. Then they have extra DNA, and that's where the plasmids come in. Now, if you remember, we use plasmids as a vector, okay? So we made use of the system so we could actually add genes to bacteria. But certain bacteria will actually have plasmids on their own, and this, unfortunately, is how they tend to get what are known as antibiotic resistance genes. So bacteria are fascinating little critters, as I've said, and these plasmids can carry different types of antibiotic resistance genes, okay? First off, they can block the antibiotic from getting in. Secondly, once the antibiotic actually gets in, it can then be altered, okay, and changed so it's no longer a threat to the bacteria. Another way is that antibiotics can degrade the enzyme. in the bacteria, they have antibiotic degrading enzymes, meaning they will break down the antibiotics, okay? They also, some of them, have what are called efflux pumps, where as soon as the antibiotic is detected in the cell, it just goes right out right away. All of these are bacterial defenses that allow them to become resistant to antibiotics, which of course is an issue for us since we take the antibiotics. Now, bacteria can actually reproduce through the process of conjugation. So bacteria can have sex the boring way, by the way. So what happens is that you'll have a donor bacterial cell and a recipient bacterial cell. That's step number one. What they're actually going to share is the F plasmid. Okay, just they call it F. It's been doing that for forever. Just realize that the donor has it and is going to give a copy of it to the recipient. So they use their pilus to connect. Okay, then the donors will end up making a second copy of the plasmid, and it'll scooch over to the recipient cell. And before you know it, the donor and the new cell both have a copy of the plasmid. Now, what's goofy about this, or not goofy, but stealthy, incredibly clever on the bacteria point, but bad for us, is this is how they can actually pass along antibiotic-resistant genes, in addition to many, many other genes. Okay, anything on the plasmid generally can get passed from one bacteria through another through this process. So this is how, bacteria can change their genome and acquire useful genes that will help them in the long run. Now, if the previous slide wasn't fascinating enough, let's talk about this one on how bacteria can actually pick up DNA from dead other bacteria. It's actually fascinating and kind of scary. So the way this happens is, let's say you have a mouse and you inject him with a type of bacteria strain that has a capsid on it. All right. Now, this bacterial species in this strain has a capsid, and because of that, it can evade the mouse's immune system, so the mouse will die. That's the first experiment that was run. The second one was to then take a second strain of bacteria, the same species, by the way, but the only difference is it doesn't have the capsid. Because it doesn't have the capsid, the mouse's immune system can recognize it and get rid of it. Therefore, even after you inject it into the mouse is healthy. Hopefully this makes sense. Okay? The next part of the experiment was when scientists took a heat-treated or heat-killed strain. Okay, this means that the bacteria was dead. So they injected that into the mouse, and the mouse is healthy. Why? Because the bacteria is dead and it can't reproduce, so it's not going to make the mouse sick. All right, now this is where the really cool part comes in. From a science perspective, not so much from a mouse perspective. You take a mixture of the second strain, okay, which is not supposed to be lethal, and a heat-killed strain of the first strain that is lethal, and you put a mixture of them into the mouse, and the mouse dies. Why, you might ask? Excellent question, I would say. The strain that was live picked up the genes to produce a capsule so it could then evade the mouse's immune system, so it killed the mouse, became lethal. Okay, how cool is that from a science perspective? Human and mouse, not so much. Science perspective, got to admit that's very cool and was a really clever experiment back in the day when it was first conducted. Now, when we talk about microbiology, you might ask yourself, well, what does that really mean? Well, you're studying microbes, okay? And basically, a microbe is anything that you can't see with the naked eye. You can imagine this is a crazy broad field, okay? So we're talking about bacteria, we are talking about some fungi, even some parasites fit into this group because many different parasites will elicit an immune response, which is kind of why they lump it into here, and they start out being invisible to the naked eye. So bacteria, viruses, fungi, parasites all tend to be lumped into the group of microbiology and immunology, which is what we have here at Michigan with regards to department. It's one of the broadest fields, okay? It's insanely broad. These guys have incredibly diverse genomes, diverse niches, diverse everything. Archaea fits into here too. You can imagine that it's kind of almost insane to lump everybody together, but that's what scientists did. Maybe it will change someday. You might also ask yourself, how do our cells compare to theirs? And I would say, aha, excellent question. So the animal cells are eukaryotes, meaning they have a true nucleus. They also have organelles like mitochondria and Golgi bodies and endoplasmic reticulum, and hopefully those terms ring a bell to you back from high school science, okay? The prokaryotic cell, now that's the group of bacteria essentially, and they have DNA, they've got ribosomes, some of them have a cell wall, some of them don't, but they definitely, they have a membrane, but they don't and are not as complex as the eukaryotic cells, okay? Yet they're still quite stealthy and they still are quite successful. So another question we need to address is what the antibiotics can actually target when you consider the differences. Well, antibiotics can target a cell wall, okay? And it's great when they target the cell wall because we don't have one, and therefore antibiotics that target the cell wall are safe for us, but not for the bacteria. Penicillin's a great example of that, okay? They can also target the cell membrane. Now, when scientists design these, you have to be really careful that they don't hurt our cells in addition to the bacteria. So this is why it's so tricky to design new antibiotics, because whatever you are administering, you have to make sure it doesn't hurt the creature that you're trying to save, that it only goes after the bacteria. So that's where it gets tricky, okay? Additionally, antibiotics can target the DNA and RNA, as well as the proteins. And remember that we have those three things too. Okay, so again, it makes it tricky when designing antibiotics, and this is part of the reason that I think corporations charge so much, because it takes a ton of research, as well it should. Okay, not that I agree with the price, but you know, some things I just try to understand. Now, through the process of natural selection, even though this is driven by humans, it's not as if we want these guys to be able to thwart our antibiotics, okay? So it is still technically natural selection, even though it's driven by humans. And what the bacteria do to overcome the antibiotics, they'll find a new metabolic pathway, okay, just through a chance mutation that allows them to be successful and everyone else dies. They can potentially bypass that part of the metabolic pathway that the antibiotic is going after, okay? And last and not least, they can also find a way to alter the drug and render it harmless, kind of like the diagram we were discussing above. Additionally, I think some bacteria can block antibiotics from getting in, but I keep forgetting to include that in this particular slide as well as the last one. Now, one of the first diseases that was actually treated using antibiotics was syphilis. Yes, ladies and gentlemen, it was one of the worst diseases back in the day in 1904. It was almost incurable, and it was just everywhere. And yeah, it was an STD, I hate to say. It used to be treated with inorganic mercury salts, okay, but that had severe side effects and it didn't work all that well. So a chemist came up with a somewhat toxic drug that did kill off the syphilis bacteria. And it took 606 rabbits to figure out which one worked, okay, because rabbits could come down with syphilis too. So eventually they did find a chemical that would, you know, go after the syphilis and kill it. It was called neosalvarsan. It was initially used quite a bit, but unfortunately it was a little bit toxic, okay, and had a lot of side effects, and eventually it was replaced by penicillin when that was discovered, okay. So, you know, it was good that that was discovered, but that's what the very first antibiotic was used for, because syphilis was running rampant all over the place. So it's interesting. Now, when penicillin was discovered, many people were happy because it was much safer than salvarsan or any of the other drugs that were eventually being used to kill off bacteria. Alexander Fleming was trying to get other scientists to realize and develop penicillin, okay? He wasn't able to do so initially. He discovered it by accident, which is interesting. He had left some bread out in the lab, which, by the way, you never do that today. And some of the bread mold landed on an agar plate, and he noticed that there was no bacteria growing around the bread mold, which told him that that particular type of mold, which came from penicillium, could actually deter bacteria. And he was correct. It goes after the cell walls. And so it took forever for him to get people to realize how useful this was, and eventually another company took it and then ran with it and made it safe for humans to be able to ingest and made it a lot cleaner and purer. And so this is how our beloved penicillin came about. Now, just on an interesting side note, my dad was actually allergic to penicillin, which you can imagine was a horrible thing back in the day, because he was alive back right when it was discovered, and everybody thought it was going to be such a great thing for everyone, and then he couldn't even take it, so he still got sick. Boo hiss disease. Boo allergies. Now, the way we test to see if an antibiotic is actually useful for killing bacteria is to do what's called the Kirby-Bauer technique. Okay, many of you may have heard this before. It's where you grow a lawn of bacteria on a plate, and then you add filtered tiny little discs that are soaked in different types of antibiotic, okay? And the idea is whoever has the largest inhibition zone, where there's the least amount of bacteria, works best for killing off that strain of bacteria, okay? For example, in the example on the right, you see A, which that that antibiotic doesn't do anything against the bacteria, whereas E has the largest zone of inhibition and probably does the best. Okay, so they don't really use this as much today. It might be used initially to see if something's promising, but it doesn't always work that way. On an interesting side note, back when I used to teach microbiology, we tried using oregano oil to see if it was actually would work against bacteria. And on the strains of bacteria we had, by the way, oregano oil worked better than bleach, worked better than cleaning products, worked better than anything. I don't know why it isn't used more. I know that some chicken farms, rather than giving their chickens antibiotics, they give them dried oregano flakes in their food. It's probably more expensive to produce than other things. It also is kind of this crazy acting oil. Like, it's really oily. You smell like a pizza, okay, when you use it. And I had it in a sealed container in a plastic bag one time. And I kid you not, it kind of flew up the container and contaminated the plastic bag. It was the strangest thing I'd ever seen. Either that or someone had dropped it and didn't tell me. But not that many people would go into that particular lab. So I just think something crazy was going on. But next time you guys are worried about, bacteria of some sort, oregano oil does work well, just a bit expensive. Unfortunately, after using antibiotics for too long and too much, we've created antibiotic-resistant bacteria. So this is where MRSA comes in. This is where resistant strains of bacteria come in, and unfortunately more and more of them keep coming about. The reason back in my day that this has been going on for a while, keep in mind, but it's getting more prevalent. I remember in the 80s, yes, I was alive. Okay. As my husband likes to remind me, I am older. But back in my day, I remember going to the doctor when I was sick, and they would send me home with antibiotics before they knew if it was bacterial or viral. Well, as much as that was a nice thought, that was actually a big mistake, because if you're taking antibiotics and it's a viral virus that's causing your disease, the antibiotics aren't going to do any good because they only go after the bacteria. But then what you do is you kill off all the good bacteria, and then you could potentially be repopulated with bad bacteria. Okay? A lot of times, too, people might take their antibiotics until they feel better, thinking they don't need it anymore. Well, then you're only killing off part of what's causing the disease, and the other one might actually come back. So that's a bad thing, too. Okay, so, this unfortunately is all caused by humans, and it doesn't just end with the antibiotics, by the way. There's other things that are not helping. Other products that we use are also not helping when it comes to the war against the microbes, and they're retaliating through antibiotic resistance. A great example of this is our antibacterial hand soap and wet ones and Lysol. And I realize we're all using these now. Okay, so we kind of all have to right now. But we might want to consider sometime whether or not it's a good thing to keep doing. Because eventually what's going to happen is we're going to kill off all the resistant bacteria, and we're going to be left with, we're going to kill off the non-resistant bacteria, and we'll be left with nothing but resistant bacteria. Then what do we do? Okay, so think of the super rats in London. All right, eventually it gets to the point where you start poisoning yourselves. Now, I'm going to offer a second potential for extra credit because I'm really curious about this. There is a product called Triclosan, Triclosan, which is a known antifungal and antibacteria product. It's very often added to mouthwashes and toothpaste and wet ones and all sorts of things. Now you might ask, why am I singling this one out in particular? Well, the interesting part of this, and slightly somewhat scary part of this, is the fact that we are consuming so much triclosan that they're actually finding it in our waste treatment centers. Okay, so it's coming out of our urine, essentially. So is that going to cause us problems? Well, I hope not. To be honest, nobody really knows. But is it good for us to be excreting a chemical that doesn't belong in our body? It's a little reason to cause for concern, I think. And so if you guys will search your toothpaste, search your wet ones, search your hand soap, okay, do a search in your cabinet and tell me if you see any of them where one of the ingredients is triclosan, and I'll give you guys extra credit. All right, because I'm really curious. So that is the lecture for today. I gotta finish grading some quizzes. Again, you all get 1 extra credit point at least, potentially 2. I want to see where the quiz average is. And they will be graded and uploaded by Tuesday before I go to bed. Your new quiz I'll release tonight, but don't take it yet. Remember, it's only going to be over lectures 5 and 6. don't take the quiz until you get the old one back to see how things went, okay? Because that'll tell you if you have to tweak anything. But I'm still going to release it in case some people are just dying to get it over with. So stay safe, everyone. I hope things are going well. And if you run into any issues, please let me know. Talk to you soon.